Hello everyone, this is Pratima and welcome back to Planet Physiology. In part 1 of apoptosis, we have seen that apoptosis, that is programmed cell death, is initiated either by intrinsic or extrinsic pathway or by perforin granzyme pathway. It is then followed by systematic breakdown of the cell. During this process, various caspases play important role. Today we shall unravel the complex mechanisms that regulate apoptosis and its clinical significance. Apoptosis is a very tightly regulated process and involves balance between numerous pro-apoptotic and anti-apoptotic proteins. Pro-apoptotic proteins promote apoptosis, whereas anti-apoptotic proteins prevent apoptosis. Since this is a highly complex process involving many genes, I will be concentrating only on the most important regulators. Okay, we have seen that caspases activation is a crucial step in initiation of apoptosis. Normally they are present in cytoplasm in inactive form and called as procaspases. In response to the stimuli, they dimerize and undergo cleavage. This dimerization and proteolytic cleavage activates them. Once the caspases are activated, the signaling cascade for cell death begins. The initiation of apoptosis is tightly regulated by activation mechanisms because once the apoptosis has begun, it inevitably leads to cell death. So let's discuss regulation mechanism, starting with regulators of caspases. In intrinsic pathway, main initiator caspase is caspase 9. It is activated by cytochrome C in presence of apoptotic protease activating factor 1. Cytochrome C is released from mitochondria in response to the apoptotic stimuli. Normally, cytoplasm also has inhibitors of apoptotic proteins, that is IAPs, and they prevent activation of caspase 9. But in response to apoptotic stimuli, mitochondria also releases SMAC, second mitochondria-derived activator of caspases. It in turn inhibits IAPs and promotes activation of caspase 9. Thus, the main activator of caspase 9 are cytochrome C and SMAC. Both are released from mitochondria. So let's study regulation of mitochondrial permeability. The most important protein to alter mitochondrial membrane permeability are the products of the gene called BCL2. These proteins are collectively referred as BCL2 gene family proteins. Based on their structural and functional similarities, these proteins are grouped into three categories. First is anti-apoptotic proteins. They prevent apoptosis and promote cell survival and include BCL2, BCLX, BCLXL and BAG. BCL2 is attached to the outer mitochondrial membrane and prevents pore formation and release of cytochrome C and SMAC. In contrast, pro-apoptotic proteins like BAX, BAK, BID remove BCL2 from the mitochondria and promote mitochondrial outer membrane permeabilization. Thus, they allow release of cytochrome C and SMAC and initiate apoptotic signals. The third category is BH3 only proteins. It includes BAD, BIM, NOXA and PUMA9. They respond to cellular stress signals and activate downstream pro-apoptotic proteins. Thus, BH3 only proteins are also pro-apoptotic in nature. The balance between pro and anti-apoptotic proteins decides the fate of the cell. BCL2 gene family proteins are regulated by another very important gene called as TP53 or just as P53. It is an important tumor suppressor gene and it is involved in various cellular mechanisms like regulation of cell cycle and apoptosis. Its protein called P53 is associated with intrinsic as well as extrinsic pathway of apoptosis. P53 induces transcription of several genes involved in apoptosis like BID, BAX, trail receptors, FAS and favor apoptosis. This action is known as transcription dependent function of P53 in apoptosis. p53 is also involved in apoptosis by transcription-independent mechanism. In response to apoptotic signals, p53 moves to the mitochondria and forms inhibitory complex with BCL2 and also to BCL-XL. This induces mitochondrial membrane permeabilization, causing release of cytochrome C. p53 also activates BOX, which in turn inhibits BCL2 and increases mitochondrial membrane permeability. Apart from p53, other tumor suppressor genes like BRCA and APC also help in regulation of apoptosis. Next is TNF gene superfamily proteins. They act as ligands as well as receptors. Important ligands include TNF-alpha and Fas ligand, whereas receptors include CD95, that is Fas, and TNF receptor 1 and trail receptors. They play important role in extrinsic pathway of apoptosis, which is crucial in elimination of infected and transformed cell by T lymphocytes and prevent carcinogenesis. Next regulators are microRNAs. Yes, the microRNAs are discovered by Victor Ambros and Gary Ruvkun, who received Nobel Prize of this year in Physiology and Medicine. MicroRNAs directly target the genes involved in intrinsic as well as extrinsic pathways of apoptosis. For example, microRNA 34 and 153 target BCL2 family genes, while microRNA 186 and 421 alter expression of caspases. Due to their various modes of actions, microRNAs are important targets in cancer treatment. The last regulators are sphingolipids. These are the membrane lipids which regulate cell proliferation, migration, adhesion, senescence and apoptosis. Important membrane sphingolipid is ceramide. It is a powerful apoptotic molecule and induces Box-mediated apoptosis in certain cancers. Their downregulation is associated with carcinogenesis. Ceramides can be converted to glycoceramide and sphingosine 1P by specific enzymes, and both these are potent anti-apoptotic molecules. Upregulation of these enzymes is also associated with carcinogenesis. Now let's move on to the significance of apoptosis, starting with its importance in normal physiological processes. First is role of apoptosis in development and maintenance of tissue homeostasis. During development phase, apoptosis helps to shape the organ and tissues by removing excess cells. For example, in formation of fingers, cells forming web undergo apoptosis to separate the digits. Another example is removal of clones of lymphocytes which react to the self-antigens during pre-processing. This helps to prevent autoimmune diseases. Apoptosis also helps to maintain health by removing damaged or mutated cells. Thus, it reduces the risk of diseases like cancer. Any dysregulation of apoptosis can lead to serious health issues. For example, mutations or mechanisms that prevent apoptosis allow damaged or mutated cells to grow uncontrollably, leading to cancer. Loss of function mutation of p53 gene fails to prevent apoptosis and allow cancerous growth of the cells. And mutation of p53 is responsible for almost 50% of the cancers. Understanding these mechanisms can lead to novel therapies that reactivates apoptosis in tumor cells. Conversely, excess apoptosis can lead to death of healthy cells. Example, neurodegenerative diseases like Alzheimer's disease leading to loss of neurons, or autoimmune diseases or even AIDS where HIV induces excessive apoptosis in CD4 cells. In such cases, controlling apoptosis could be helpful. Now let's see therapeutic implications of this knowledge. Understanding apoptosis opens new avenues for treatment. Targeting apoptosis in cancer treatment is the first one. Targeted therapy aims to make cancer cells more susceptible to apoptosis. Similarly, strategies to inhibit excessive apoptosis may help protect healthy cells and neurodegenerative diseases like Alzheimer's. Thus, understanding mechanism of apoptosis and its regulation can lead to significant advancement in medicine. Coming to the last part of the session, indicators of apoptosis. First is DNA laddering. DNA fragmentation is considered as the hallmark of apoptosis, which is caused due to activation of endonucleases leading to cleavage at 180 to 200 base pairs. It can be easily visualized by agarose gel electrophoresis in the form of DNA ladder. As shown in the picture, laddering is present in second and third sample but not in the first one. We can also perform annexin 5 assay, which binds to phosphatidylserine. Since this protein gets externalized in apoptotic cells, annexin 5 assay can easily differentiate between apoptotic and necrotic cell death. Next, we can easily assess expression of apoptosis regulating genes by RT-PCR or their protein products by Western blot method. Most routinely assessed proteins are BCL2, BOX, caspase-3, 8, 9 and P53. Another important test is to assess mitochondrial membrane potential. One of the methods is by using JC1 probe and fluorescence microscope. Decrease in the ratio of red to green fluorescence indicates loss of mitochondrial membrane potential. Usually, increase in BOX to BCL2 ratio induces pore formation in the mitochondrial membrane. This causes loss of membrane potential, leading to release of cytochrome C and initiation of apoptosis. Thus, to summarize, we have seen that apoptosis is necessary for normal development of the tissues and at the same time to remove the damaged or mutated cells. Thus, it is a vital process to maintain health of the tissues and hence the person. Activation or inhibition of apoptosis is a very tightly regulated process and involves multiple genetic mechanisms. Most of the processes are directed to regulate activation of caspases. BCL2 family proteins, P53, TNF gene superfamily proteins, microRNAs and sphingolipids like ceramides play important role in controlling apoptosis. Dysregulation of apoptosis can lead to diseases like cancer, Alzheimer's or AIDS. So that's all for this session. Thank you for watching and see you in the next video. Are you new to my channel? Then please subscribe it and press the bell icon to stay updated about the new releases. If you like the video, give it a thumbs up and share it with your friends. Thank you for joining in and see you in the next video.